Hi, I'm Ding from COPSAC. Our project is Can neurodevelopmental disorders be prevented in early life toward a precision prevention platform? Neurodevelopmental disorders, including ADHD and autism, affect up to 1 in 5 children and represent a major lifelong burden for families and the healthcare system. Yet no validated primary prevention strategy currently exists. Our project explores a potentially disruptive idea in preventive neuroscience that NDD risk may be modifiable during critical early life windows through responder-guided intervention. In other words, instead of waiting until symptoms emerge, we ask whether we can identify biological risk and response profile early enough to enable prevention. Our preliminary randomized control trial findings provide an encouraging proof-of-concept signal. In a genetically defined subgroup representing approximately 50% of our cohort, prenatal facial intervention and elevated maternal CNPF-related metabolites were associated with 44 to 86 lower order of NDD risk in offspring. This is important because it suggests that inconsistent results from previous prenatal intervention studies may reflect biological heterogeneity rather than lack of efficiency. The implication is potentially transformative. Instead of one-size-fits-all approach, we may be able to identify biologically defined responder groups, target intervention to those most likely to get benefit, and develop alternative formulation for predicted non-responders. We are now building a broader precision prevention platform. First, we will validate responder biology across 3 deeply phenotyped mother-child cohorts worldwide using longitudinal multi-omics data and neurodevelopmental outcome. Second, we will investigate the underlying neuroimmune and immune metabolic mechanism using hepatocyte microglia system and translational piglet models. Third, we aim to translate these findings into a companion diagnostic workflow capable of identifying likely responders early in life. Importantly, we are also exploring next-generation neuroprotective formulation for predicted non-responders. This expands the project from biomarker discovery toward a scalable intervention platform. We already have strong clinical cohort and mechanistic foundation in place. A key strength is access to one of the world's most deeply phenotyped mother-child cohort with randomized intervention data and longitudinal neurodevelopmental follow-up. We are now looking for collaborators who can help us translate this concept toward a scalable precision prevention. In particular, we are interested in companion diagnostic, formulation development, translational neuroscience, and cumulation pathways. If successful, this could establish a first-in-class framework for preventive neuropsychiatry. The long-term vision is to move from reactive treatment toward early biological risk modification in neurodevelopmental disorders. The platform may create opportunities around companion diagnostic, responder-enriched intervention, and future commercialization in maternal child health care. Thanks for listening.